Hi, and welcome to this lecture on environmental adaptation of animals. On this series, I'm going to describe the molecules and cells in this animal physiology. So, something that you should already know. And now we are considering on a single cell type, epithelia. Well, why we are considering on epithelia? Well, it's because it's the cover of this whole animal. It's also covering all the organs inside this animal. And it covers all the cavities in these animals. So if we have an organ and there's a blood vessel, the blood vessel is covered by the epithelia. And it's actually then there's the border shown between the interior of the animal and external environment of the animal. And so that's why, depending on what goes through this epithelia, it's depending giving the idea that, okay, what is affecting on these animals. So it's limiting the access of these molecules. And for example, in previous video, I was showing that, okay, we have snakes, we have poisons that are affecting on prey. But if the prey has certain kind of epithelia that is limiting the access of this drug or this poison in this inside in the interior of the animal, then it's not affecting. But then, on the other hand, it's also to potentiate the access of molecules. So if we have a very tight epithelia everywhere, we can't get any food particles inside in our body, etc. And it's classified by the shape. It can be flat, cube, or column. So the shape of the cell. Over here we have column type because it's on kidneys. And then it can be classified by the thickness of the tissue. It can be simple, like over here we have one single cell layer of cells, but then it can be also stratified, so there are several layers of cells in that case. And if we think on what is the shape of the epithelia, it's that, okay, the cells are touching each other. There's not much stuff between the cells. And there are two kinds of surface. There's apical surface facing out from the tissue and basal surface that is facing toward the tissue. And these are two completely different kind of membranes. And in the shape can be different. So on the right hand side we can see that, okay, these are cubic because it's as the width and the height is similar, but on the right hand side in internal epithelia in our intesticity, it's more like column, so it's a little more longer one. And it can be increased the surface by having curves, likely in microvilli. And then on the other hand, other side, there can also be produce some granules to secrete some material. So it can be some endocrine cells. And the shape of the epithelia has some kind of meaning. So in the nephrons, we have a lot of surface, so a lot of microvilli inside it, so to increase the absorption of material. And then there are a lot of mitochondrials to give enough energy for the cell processes, etc. But then when we have a blood vessel, this is very flat cells, and so there's an easier flux of gases from the blood vessel to tissue, etc. And actually they are so flat, so that's why, although over here we have three different epithelial cells, so there's only one nucleus seeing, so it's a little bit longer cells. So because if the nucleus of all these three cells would be in the same place, there would not be enough space for the red cells to pass through this lumen. And the regulation and through the epithelia is based on the cell junctions. We have different kind of junctions, so binding these epithelial cells together. First of all, we have tight junctions that are common in some basic level causes. They are in vertebrates, and in invertebrates there's quite often these separate junctions. They're doing the same thing. They're gluing these cells together and then limiting the access of particles to flow between these cells. And then we have different kind of junctions that are making stronger contacts or interacting the cells. So now particle that is in one cell can be transported to other cells, etc. And these junctions are important to maintain the membrane protein pool in apical and basal side separated. So quite often in epithelia there is an apical region of this cell membrane, and the basolateral region is separated by a band of tight junctions. So they go completely around the cell, and there's no diffusion so the membrane protein that is in one side, it can't just flow to the other one by this membrane. And that's why these regions are different in the protein components. And then how the molecules can be transported through this epithelia. Well, first of all, it must cross both the apical and basolateral membrane quite often. So we can have absorption with increase the absorption with the microvilli, and there are also some transporters that are actively taking material from, for example, in our intestivity, and then transport it in on the other side and to the blood vessel. But in very small molecules, they can flux between the epithelial cells. And then in action, it's why the epithelia is important. It's important because otherwise we can't understand kidneys, for example, at all. So it's very important to understand that, okay, we have two different parts on this. And for example, in kidney early proximal tubules, so solutes are moved together with sodium. So here you can see in the top it's H2O, so meaning that, okay, there's aquaporins transporting water from the apical side to the basal side. Then a little bit lower there is some carbon dioxide that is transported. Then we have sodium and sodium proton exchanger that is transporting sodium. Then there is sodium together with phosphate, glucose, amino acids, organic anions, etc. that are transported on the apical side. And then on the other side, there are some other transporters. And it's very important that we have different transporters on one side and another side. Thank you.